So today we are going to learn insulin secretion. Now, first thing you need to understand is this: why this chapter is important, or why this topic is so much important. The first thing you need to understand why this is important so much is that insulin secretion, or the mechanism of insulin secretion, actually contains many drugs which you will be using. Okay. Now, the first point you need to understand is this, that when you are talking about insulin secretion, we'll discuss actually, whenever you are talking about insulin secretion, clear, insulin secretion, the first thing you need to understand is this, that insulin is secreted and produced in pancreas or pancreatic beta cell. Okay. Now, in pancreatic beta cell. Okay. Now, a very important point you need to understand is this, that suppose this is your pancreas. Okay. How will you know that this is pancreas, this is not, this is not salivary gland? Because the ducts are approaching the assignee, right? Now, a very important point you need to understand right now is this: as ducts are approaching assignee, that means it's a pancreas. Now, in between these two assignees, suppose there is a cluster of cell, that's why it is called islet of Langerhans. Langerhans discovered it, and that's why it is called islet of Langerhans. Islet means island, or here it is island of cells, islet of Langerhans. Clear? Islet of Langerhans. Now, a very important point here you need to understand is this: in the beta cell, okay, beta cell actually, suppose this is your islet of Langerhans, this is your islet, okay, and beta cells are present almost in the center. Beta cells are, in most of the cases, beta cells are always present in the center. That is, beta cells are always present in the center, which is surrounded by alpha, and they are surrounded by alpha cells. Okay, they are surrounded by alpha cells, and delta cells can be anywhere. They can be, here, they can be here, they can be here, anywhere. Okay. So, this is the arrangement of cells in islet of Langerhans. A very important point you need to understand is this, that if suppose beta cell is secreting insulin, then alpha cell will also know that insulin has been secreted by beta cells. That means every cell knows what other cell is secreting in islet of Langerhans. Okay. Now, let's cut short everything and talk about insulin secretion. Okay. We are not discussing this because we have already discussed how a pancreas looks like. We have actually done the details of every anatomy. Now, what is insulin secretion, right? Now, very important point you need to understand is this, that suppose this is your pancreas, and this is suppose your beta cell, and we are taking it and we are actually magnifying it, and suppose this is your beta cell. Suppose this is your beta cell. This is your beta cell, a very large magnified version of beta cell. Suppose this is your beta cell. A very important point you need to understand right now is this, that if you want to learn insulin secretion from beta cell, you need to understand three basic things. Okay. That is, there are already, vesicles which are loaded with insulin C peptide. Okay. there are already, vesicles within the beta cell of pancreas which are loaded with insulin and C-peptide. Okay. Now, another thing you need to, this is the first point, you need to understand that beta cells are loaded with insulin. Clear? This is the first point. Second point you need to understand is the structure of potassium channel. Okay, the structure of specialized K plus channel. Now, this is a very important part because pharmacological, application will be there. Now, why do you think that we are discussing a potassium channel? We have discussed many channels, okay, but why we are actually especially discussing this potassium channel here? This is because this potassium channel is unique here. Now listen, suppose this is the cell membrane, and now we are going to discuss the potassium channel which will be present here in the cell membrane of beta cell. Now listen with all your attention. Every potassium channel in this world looks like this. Okay, wait. Every potassium channel in this world looks like this. This is suppose your every potassium channel looks like this. Okay, one, two, 3, 4, 5, 6. Okay, almost every potassium channel looks like this, and there are other domains associated with it. Okay, almost every potassium channel looks like this. This is the N terminal, this is C terminal. Now listen, N terminal and C terminal of potassium channel are actually inserted inwards. Okay, now there are many other channel, sub part of this channel. Okay, subunits of this channel. We are not talking about this now. A very important point you need to understand is this: the first four subunit of potassium channel is absent in these type of specialized potassium channels which are loaded in the beta cell, right? That means their structure looks like this. Their structure looks like this. Clear? Their structure looks like this. This is your potassium channel. Okay. Now, very important insight into this structure is this, that this is the lipid bilayer. Okay. This is suppose the lipid bilayer. Okay. Now, this is the lipid bilayer. Now, a very important point you need to understand is this, that this is actually the uniqueness of this potassium channel. It's true, but there is another thing which is associated with this. You know what? That is there are specialized domain attached to this potassium channel. That means these domains are not at all present in any potassium channel which are present in your body. That means these are specialized potassium channels which are loaded onto the membrane of beta cell of pancreas. Now, what is the name of these domains? Right? These are called SURs. These are called SURs. Or what do you mean by SURs? Sulfonyl urea receptor. Now, SURs, sulfonyl urea receptors. What do you mean by sulfonyl urea receptor? Now listen. The meaning of sulfonylurea is actually, it's a drug. It's actually a drug which is associated with the inhibition of these receptors or this domain. Okay. So what is sulfonylurea? Sulfonylurea actually inhibits this domain. Okay. Now, another third point you need to understand, a very interesting part about this, that is carbohydrate hormones, neurotransmitters. These three, carbohydrate, these four, amino acids, carbohydrate, amino acids, hormones, neurotransmitters. These four actually control the insulin secretion from beta cell. Clear? These four control the, insulin secretion from beta cell. Now we will talk about, insulin secretion in details. Now, very important point you need to understand here is this, that is, suppose this is a beta cell. Okay. Now, if I tell you what is this, then you will be able to answer me, right? This is potassium channel. Now, the specialized name of these potassium channels are KIR 6.2. What do you mean by this? I will tell you later. Now listen. Now, this is the name of this channel, KIR 6.2. Okay. IR means what? IR means inward rectification. IR here means inward rectification. Inward. That means what? Rectification means where the potassium ions will move. Okay. Now, a very interesting part you need to understand is this, though it is inward rectifying, channel, but actually potassium moves out. Now, what is this domain? What is this domain? This is sulfonylurea domain, right? Or sulfonylurea receptor, right? Now, a very interesting part here is this. A very interesting part here is this. Potassium channel is continuously, suppose, moving out of the cell through this KIR 6.2 receptor, okay? Through this potassium channel receptor. This is the potassium channel. So, first thing you need to understand is this potassium is now moving out. This potassium is now moving out. Now, because of this cells are supposed to keep a high concentration of what? Cells are supposed to keep a high concentration of potassium. Everybody knows that, right? Cells are actually, go downs of potassium. Cells should actually overload potassium. There is no problem in it. Cell can actually, load potassium more. But when these potassium ions are moving out, that means they are losing positive charges. They are losing positive charges are moving out. And when positive charges are moving out, cell cannot be depolarized. Depolarization is important, but in this scenario, this cell cannot get depolarized. Okay. Now, what you will have to do is this. You will have to, stop this potassium channel outflux or efflux, efflux in some way or the other, right? Now, what is your first, approach? That is, suppose you take, that whenever in your diet carbohydrate concentration is high, insulin concentration also becomes high, right? Clear? Now, what happens? This is GLUT2 receptor, okay? Now, when glucose, suppose you have taken a high carbohydrate rich meal and this glucose comes into the cell or beta cell. Then and there it is converted into glucose 6-phosphate by the help of which enzyme? Glucokinase or hexokinase, right? Glucokinase or hexokinase, right? Then and there it is in. Now this glucose 6-phosphate will enter the glycolytic pathway and give out 2 ATP, right? They will give out 2 ATP. Now important point here is this, suppose this is your pyruvate, this pyruvate will also now move into the what? Mitochondria. Pyruvate will also now move into the mitochondria. They will be converted into acetyl coenzyme A, and acetyl coenzyme A together with oxaloacetate will enter TCA cycle, right? They will enter TCA cycle. Clear? They will enter TCA cycle, and again due to the entrance of this acetyl CoA there will be another what? ATP concentration will increase in the cell, right? Now you need to understand a very basic thing, that you want to end this potassium efflux. Now a very important point, that means you will have to block this particular channel. To block this particular channel, what you need is ATP. ATP actually inhibits this. Okay, ATP actually inhibits this. ATP inhibits this. Clear? ATP, whenever ATP levels are high, ATP or NADPH levels are high, whenever ATP is to ADP levels are high, that means ATP concentration is high, then K plus channel will not work. That is their ATP dependent potassium channel. That means if ATP is increasing in the cell, then they will stop working. Clear? Now, a very important point you need to understand is this. So, when ATP is increasing, then these potassium channels will stop working and potassium efflux will stop. Due to the stoppage of this potassium efflux, what will happen is very important. Now listen. Therefore, the cell will undergo depolarization. Okay, that means due to the stoppage of this efflux, the result is depolarization. Depolarization means what? Depolarization means what? Now listen, it's very important. I have told you already, but it's okay. Now listen, just for your recap. Inside the cell there will be negative charge, outside will be positive charge. Why? Because there are more cations outside the cell, and these negative charges inside the cell is because of proteins. Now listen, due to some disturbances, if these positive charges enter, then they will become positive, and with respect to this positivity, they will become, the outside will become negative. That means this was like a battery. This was like a mini battery, and now the polarity of this battery has been reversed. That's why depolarized. Now, important point you need to understand from biological point of view, depolarization or depolarized means excitation. Clear? Excitation. Depolarized or depolarization means excitation. Now, whenever I am talking about that, when this potassium channel will be inhibited, then this will lead to depolarization. That means cell will become excited. Cell will become excited, and when this excited cell, when this cell will be excited, then a very important point will occur. That is, there is a channel here, you know. This channel is called voltage gated or sensitive, voltage sensitive calcium channel. Clear? This is called voltage sensitive calcium channel or voltage gated calcium channel. Now, due to depolarization, there will be disturbances in your, you know, potential differences. There will be, you know, different potential difference across different regions of the cell membrane. Therefore, there will be different voltages across different cell membrane areas. Therefore, these potential difference changes will be actually sensed by this voltage sensitive channel, and calcium will start their influx. Calcium will enter. Now, when calcium is entering more and more, when more and more calcium is entering, suppose this is your, you know, channel, okay, suppose this is your calcium channel. When more and more calcium ions are entering the cell, then what happens is, it's a very interesting part, what happens is, this calcium channel actually induces smooth endoplasmic reticulum to release calcium. Now listen, what happens is, due to depolarization, voltage sensitive calcium channel opens, and when this calcium channel opens, more and more calcium enters the cell, and due to this, smooth endoplasmic reticulum now start calcium release. This is called calcium induced calcium release. Okay, this is called calcium induced calcium release. Calcium induced calcium release. Okay. Now, this is calcium induced calcium release, right? Now, after that, what happens is very again interesting. I will have to, you know, make this diagram a little bit big. Okay, I can manage here. Now listen. After that, what happens is, these calcium ions which are actually coming out of the, you know, smooth endoplasmic reticulum, they will actually bind with calmodulin. You know what is a calmodulin? If you don't know, then I should say that you revise GQ signaling, okay, or smooth muscle contraction, which is actually same. Now listen, almost. Now listen, this calmodulin protein, you know, have 4 pockets, and in those 4 pockets calcium will sit. In those 4 pockets calcium will sit. Okay, calcium will sit, and this calcium, 4 calcium together with calmodulin complex will activate another protein called CAM kinase. Okay, calmodulin kinase or CAM kinase. Now, what will this CAM kinase do? Right? Now listen. I don't remember where they have written that it actually, you know, phosphorylates those protein associated with synaptic transmission. It was written somewhere. Don't talk rubbish things. In a 2015 paper, which is there in your NCBI site, you will understand what happens. Okay. The most important thing you need to understand is this: whenever any kinase, during insulin secretion at least, whenever any kinase gets excited or whenever any kinase is activated, then a very important and a very peculiar thing happens. Now, what happens is, here there is, you know, actin myosin filaments. Now, due to these CAM kinases and calcium, these actin myosin actually start their movements. These actin myosin actually start their movements, and what happens is, suppose this is your actin myosin filaments, okay, actin and myosin filaments. Now important point to understand, suppose this is your insulin plus C peptide containing vesicle. Now due to this, these will come here and they will try to bind with V-snare, T-snare. But don't ever tell these rubbish things that V-snare is coming in contact with T-snare because of phosphorylation from CAM kinase. This is not true. CAM kinase what actually does is actually it excites actin and myosin in such a way so that they can start their small movements, leading to the release of, or leading to actually the movement of this particular vesicle towards the active site. If you don't know what is active site, go to the video where I have explained synaptic vesicles or synaptic transmission. Now they actually help in the movement of this vesicle to the active site, and there they actually combines with V snare and T snare, and after that what happens, and if you don't know what happens after that, go to that video where I have taken the class of synaptic transmission. You will understand. Okay, clear? Okay, now so this is the basic part. This is how your glucose or your food particles control insulin secretion. But I have told many things, right? How amino acid controls. Now it's very important. Amino acids, I can tell you that leucine, this amino acid suppose has a channel and it enters into TCA cycle. Okay, it enters by changing itself, it enters into TCA cycle, and therefore ATP concentration of ATP increases. Therefore concentration of ATP increases and potassium channel is blocked and depolarization takes place, leading to voltage sensitive calcium channel opening and all these things. Okay, now how any neurotransmitter can actually help? How any neurotransmitter can actually help? Now, a very interesting part you need to understand here is this. If you want to know really how a neurotransmitter can help, I will talk about a single neurotransmitter, or two actually, that is adrenaline and acetylcholine. Okay, now if you want to really talk about adrenaline, you must understand that adrenaline binds to adrenergic receptors, right? They binds to adrenergic receptors, right? Now there are two types of adrenergic receptors, right? Alpha and beta. A very important point you need to understand is this, that if you want to learn the effect of adrenaline on the insulin secretion, you will have to understand this, that if you want to really learn the effect of insulin secretion, effect of adrenaline on insulin secretion, you will have to first understand that whether adrenaline binds with alpha receptor or beta receptor. Why? Because a very important point you need to understand is this. Suppose this is beta receptor. Suppose this orange color one is a beta receptor. Beta receptor is coupled to GS, and you know what will happen. You already know what is cell signaling. GS. From GS, what will happen is protein kinase A will be activated and it will similarly work like CAM kinase. Now, a very interesting part you need to understand is this. Suppose this, okay, let me draw with identical colors. Suppose this is your beta receptor and this is your alpha receptor. Okay. You also know this, that whenever we are exercising, we are in a stress, that means exercise is a stressful activity. Now during stress, S for stress, S for sympathetic activation, that is during stress sympathetic system will fire and releases adrenaline. Now a very important point you need to understand is this, that if all the beta receptors are activated, then what will happen is this, you need to understand is, suppose all the beta receptors are activated, then that means what? Insulin will go out, more amount of insulin will go out, right? More amount of insulin is going out means what? A rapid decrease in blood glucose level will occur, you will fall, you will have a blackout. Okay, suppose you have gone to a gym and in front of a girl, just you have taken 2 dumbbells and you have a blackout. Probably it's the right time for you to leave the gym. Okay, now important point you need to understand here is this, that's why there was a study that showed beta cells have both alpha and beta receptors, but the concentration of alpha receptor is more. Clear? Why you know now that? Okay. Now what about other things? What about acetylcholine? Because we need acetylcholine. Acetylcholine is actually secreted when we are at a resting stage. That means when sympathetic nervous system is actually firing. Okay, when sympathetic nervous system is actually firing, we are at rest, and due to this resting condition, sympathetic nervous system fires. Parasympathetic nervous system fires, and parasympathetic nervous system is what? Cholinergic system, and cholinergic system means what? It will release acetylcholine. That means we should have a receptor for cholinergic receptor. Suppose this is your acetylcholine receptor. It is not no, suppose. Now important point you need to understand is this is coupled to GQ. Now acetylcholine binds here, this is coupled to GQ. Cholinergic receptors are coupled to GQ. Clear? So that means whenever I am talking about GQ signaling, phospholipase C will be activated and all these things, but after that, after all these things, what will happen directly? CAM kinase will be activated and the similar things will occur. Clear? The similar things will occur. Clear? Okay. So now you know how adrenergic system works or helps in the insulin secretion, and how parasympathetic system works or helps in the insulin secretion. Now let's talk about a hormone, specific hormone, that is CCK, cholecystokinin. Cholecystokinin is a hormone which is secreted from eye cell of intestine. Now important point you need to understand is this, we have discussed in detail cholecystokinin in our GI class of alimentary canal. Now important point you need to understand is this, that this cholecystokinin which is secreted from eye cell actually have a similar type of receptor like that of ACH. That means they are receptors, the receptors to which cholecystokinin binds, they also are coupled to GQ. Ultimately they will activate CAM kinase. Clear? Okay. Now we will be talking about a clinical implication of insulin secretion. What is this clinical implication? Do you know what is glimepiride? You know what is glimepiride? It is used by doctors when somebody is having non-insulin dependent diabetes mellitus, that is NIDDM. Okay, that means Type II Diabetes Mellitus. It's a drug. When someone is suffering from NIDDM, they are given this drug. A very important point you need to understand is this, what is the use of this NIDDM? What is the use of these drugs which actually act in NIDDM? Now listen. If I tell you how it works, you will now laugh at me because you already know how it works. Now listen. Have you ever heard of the term sulfonylureas? Not receptor, sulfonylurea. Sulfonylureas are class of drugs. Sulfonylurea are class of drugs which are very, very important for the management of type 2 or type 2 diabetes mellitus or non-insulin dependent diabetes mellitus. Clear? Now, in non-insulin dependent diabetes mellitus, what actually happens is, you know what is diabetes mellitus? Diabetes mellitus is nothing but, in most of the cases, is nothing but an autoimmune disease. Okay, it's autoimmune destruction of a beta cell. Okay, this is type 1. Now, if you have insulin resistance, that means suppose I am a cell which should be responsible to, which should respond to insulin. Suppose I have a receptor here. Okay, I have a receptor, and suppose I have a receptor, and suppose this is insulin. My hand is insulin. Suppose insulin is coming, but my receptor is not getting activated. Now, what will happen is this. That means in those patients, you know what is happening. Suppose I am a cell and I have an insulin receptor and insulin is coming. That means what? My beta cells are okay, but my own body cells are not actually, recognizing insulin concentrations. That's it. So you need to understand a very basic thing, that in those patients which have this type of thing, that means their body cannot actually, recognize insulin, or they cannot actually recognize the, normal concentration of insulin. This type of, scenario is called insulin resistance. This type of scenario is called insulin resistance. We have a whole, video library on diabetes mellitus, don't worry. Okay, this type of scenario is called insulin resistance. Now, a very important point here you need to understand right now is this: during insulin resistance, or whenever a doctor actually understands that, we'll talk about this diabetes mellitus when we will have a detailed pathology, okay, pharmacological classes also. Now, a very important point you need to understand right now is this, that whenever a doctor understands this is a case of NIDDM, so what you will have to do? You will have to increase the insulin concentration, but normally your insulin concentration will not, go high or rise very high. So you will have to do it with the help of drugs. Now, sulfonylurea was discovered in 1943 or 1940s, but still it has been used. Why? Because sulfonylurea have two generations of drug. This is the first generation. There is first generation and there is second generation. Now, the most preferred of the sulfonylurea drugs are not first generation, not the first generation. First generation is not, preferred drug, but second generation sulfonylureas are preferred drugs for, doctors right now. Say one of the best, sulfonylurea second generation drug is known by the name of glimepiride. Okay. Now, what this glimepiride does is, glimepiride actually, what it does is, inhibits this sulfonyl or this SURs or sulfonylurea receptors. They actually inhibit this receptors, and due to this, no movement of potassium takes place. That means they are working similar to that of ATP, right? Now, what is the importance of this glimepiride then? Suppose if you take more amount of carbohydrate, now listen, if you take more amount of carbohydrate, we can generate more amount of ATP. We can, effectively, stop this potassium channel. We can effectively increase our insulin concentration, but that doesn't happen like that. If we go on increasing, our calcium levels, a dangerous thing will occur, and that is, you will learn about that. Okay, but these drugs actually inhibit directly this channel. These drugs inhibit these potassium channels, and due to this glimepiride or sulfonylureas, this potassium channel gets blocked, and potassium will now not leave the cell, and therefore depolarization will occur, and after depolarization whatever I have told, everything will occur. Okay, that means CAM kinase is getting activated, and vesicles are released, and therefore in concentration of insulin inside your body, having non-insulin dependent diabetes mellitus, in those patients having NIDDM, their insulin concentration within the body will increase after the application of these drugs. This is second generation sulfonylureas. Sulfonylureas bind to sulfonylurea receptors present in the KIR 6.2 potassium channel. Okay, so this is a short video with respect to these drugs. We'll talk about them and their structure in details, how they bind. Okay, how we have studied that, what is the affinity of these drugs. We'll talk about them, don't worry. But right now, that is all for today. So you have learned insulin secretion and you have learned a bit about the drug. Okay, thank you.